Hello everyone, I am back with our new video, which is on the topic of coordination and response, which is chapter 13. So in this topic you guys need to know these 5 important definitions that include sense organs, synapse, hormones, gravitropism and phototropism. Alright, so one of the most common definition that they usually ask is sense organs and hormones. Like these are like a very popular definition that they usually ask if you check the past papers. So let's read it out together, the definitions. So we have groups of receptor cells that respond to specific stimuli such as light, sound, touch, temperature and chemicals. And the next thing we have is synapse, a junction between two nerve cells consisting of a minute gap across which impulses pass by diffusion of a neurotransmitter. Hormones, a chemical substance produced by a gland carried by the blood which alters the activity of one or more specific target organs. And then gravitropism, a response in which a plant grows towards or away from gravity. The last one is phototropism, a response in which a plant grows towards or away from the direction from which light is coming. So don't be confused between gravitropism and phototropism. So it's very easy to understand because it's literally related with gravity. So the plant always grows in response to the gravity. It either grows away or it grows towards. And under gravitropism you have the negative gravitropism and the positive gravitropism, and then the phototropism as well. So in phototropism it looks confusing at first, like how is this related to light. So just remember that you know the word photosynthesis. So since photosynthesis involves light from sunlight, so they basically included the word photo plus tropism, so it makes phototropism, and this is a response towards the light or away from the light. Okay, so let's move on to the exercise, which is exercise 13.1, caffeine and reaction time. So let's read the text in the box. You should be getting quite confident at planning experiments by now, so there are no reminders here about all the different things you need to include. You'll find it quite tricky to control variables in this one. You may be able to try out your experiment when you have planned it. So since I'm going in a very random order for answering the chapters from this workbook, so they have actually mentioned about the various kind of controls: independent variable, constant variable, dependent variable, and the apparatus, the methods. This is a specific way of answering questions that are related to experiments actually. So let's read out the first question. Reaction time is the time between receiving a stimulus and responding to it. Plan an experiment to test this hypothesis. So what's the hypothesis? Consuming drinks containing caffeine decreases reaction time. Alright, so basically since it's about hypothesis, you guys need to state what's the effect. Like since it's about hypothesis, you basically have to either prove that the statement is accurate or it's true, and if the statement is wrong, you need to show how it is wrong actually. So this is how it is. So let's look at the first point over here, which is caffeine intake. So the first thing you need to, since it's about caffeine, let's highlight this first. Since it's about caffeine, so everything that you want to try or test the experiment on, it's entirely related to caffeine. So in this case you should say that caffeine intake is varied. That means this is going to be the independent variable, because this is the one that is supposed to be changed by you. That's why the caffeine intake is actually different. Like it varies on the amount of coffee or tea a person consumes actually. So caffeine is not just present in coffee, it can also be present in soft drinks as well, but that's usually rare. But you should also, but if you want, you can also use a range of coffee concentrations, since different brands of companies use different caffeine concentrations in their coffee or tea. So like a better accurate statement that you could write about using a range of caffeine concentrations. Right, so the next thing is what are the constant variables? So among the constant variables actually include volume and the concentration. Let me just make it a different color. So volume and concentration of caffeine in the liquid drunk, and then time between drinking and doing the reaction, time test, time of the day, the age and the gender of the person, what the person has done just before the test is carried out. Basically, sometimes the reaction time can also affect what the person has eaten or drunk before, or what activity the person did before consuming the caffeine, and how many times the person has done a reaction time test before. So you need to understand that it's not possible to control all variables, but when you're answering your past paper questions, to be on the safer side, you just only have to mention 2 constant variables, actually 2 to 3 constant variables, but it should be minimums 2, and if you want to be on the safer side, you can mention 3 as well. And then the dependent variable is the one that's being affected by the independent variable, and that includes this: the reaction time measured using a stated method. For example, stated method meaning you can either use a test on the internet or catching a drop ruler. This is actually not that familiar. This kind of method, you guys may have not learned about this, and this rarely, like really rarely, it actually appears in the past paper. Normally when they ask you to measure anything related with time, they usually have to talk about stopwatch, but in this case, since it's entirely new type of method, it's good for your knowledge as well, in case if they try to make the experiment questions hard in your paper 6, then you can mention about for using a test on the internet and catching a drop ruler. And then repeats done. So now the next part of the experiment is how are you going to make your experiment reliable. So this is a very important factor that you have to consider. So one is this: repeating the experiment twice or thrice, okay? And if you use different people, then it will actually improve the accuracy as well, because one person may have the same reaction time even though it's given a different range of caffeine concentrations. So that experiment that you might have conducted will be just on one individual. However, a lot of thousands of people consume caffeine, so to make the experiment more reliable, it will be a better option for you to mention about using like, you won't say using people, but to test on different people on the reaction times. But if you want to, you can mention this, but the most important factor on how to make the experiment reliable is repeats done. Repeat the experiment twice, and if there is data given, for example, if there is if it's something to do with time, then repeat the experiment twice and take average or find or calculate the mean out of it, right? So then since it's actually a hypothesis, and according to your new syllabus, it's not really new, but normally in paper 6 they don't really ask you to draw any chart or any sort of graph thing. This is only in physics that they ask you, but in bio they don't ever ask you to draw these things in your paper 6. But there are actually graph questions in paper 6, but it's just a separate question. It's not connected with the experiment things. Usually it's not connected. So but in this case, since it's testing a hypothesis, if you have space it'll be a good option, but it's actually not compulsory for you to do this. So all in all, what are the things you have to include? Okay, so since it's a hypothesis, okay, you also need to mention what are your apparatus and what is your method of conducting the experiment actually. So that one thing you have to mention is the apparatus for measuring the caffeine concentrations. You could say using a syringe, or sometimes since you could use in a balance mass. And then the next one, which is the method. How are you going to conduct this experiment out? You're going to test it on, you're going to use different region of caffeine concentration, test it on one person, and then calculate the reaction time using a stated method. So this is actually, you need to mention all that. So it's actually mentioned over here, so you can consider this to be the method as well. So this is the method carried out. So you know that one order that's easier for the examiners to understand, you should write in this way. Apparatus, number 1 being apparatus, number 2 being the method, number 3 being mentioning independent variable, dependent variable, and constant variable. And then you have how you're going to make your experiment reliable, and it's usually this. In bio you only have to mention these things. So independent variable you only have to mention 1, dependent variable also 1, but constant variable, to be on the safer hand, it's better to mention between 2 to 3. And how you're going to make experiment reliable. Alright, so this is a very good structure of writing an answer to an experiment question, as it makes the steps and everything much more clearer. So let's move on to the next part, which is exercise 13.2. Oh God. So accommodation in the eye. So doing this exercise, preferably without looking anything up, will be a good test of how well you understand how the eye changes in order to focus on objects at different distances, which is called accommodation. Use a ruler to draw the light rays on your diagram, and take great care to show clearly where they change direction and where they are brought to focus. Right, so now the diagram shows an eye focus on distant objects. So as you can see, this is the structure of the eye, and this is the object. It's a very weird object, but this is how it is. So you can see the light is focused on retina. This is the retina, this is the cornea, and this is the lens over here. So this is how the light is actually diverging from a distant object. Now complete the diagram below to show the eye when it's focused on a nearby object. Add labels to match those on the first diagram. So we basically have to use these same labels, but the position of these labels is actually different now that the object is actually in the center. So if you look here, since the object is in the center, so how your diagram needs to be like, it needs to be like this. So this is how it's supposed to be. So as you can see, you can see that the lines, they're actually being diverged at a much shorter distance compared to the diagram over here. The lines are much more longer, the light rays especially. The light rays are more longer when the object is at a distance, but over here the light rays are actually shorter, but they diverge really big. So in this way, the cornea bends the light rays, and when it bends the light rays, the light is focused on the retina, and the fat lens bends light a lot. So you know, this actually you need to remember what happens to the cornea, what happens to the retina, what happens to the lens. Actually not retina, you need to focus on what happens to the lens and what happens to the pupil and the cornea when an object is at a distance or when it's closer, right? So let's move on to the next part. Describe how the changes that you have shown are brought about. So use these words in your description. So now the question is okay, you know, the way how they structure the question, the workbook is very vague actually. They don't really do like this in your paper for biology question paper. It's always clear. It's just in the book it's kind of unclear. To some people it may be unclear. So how the changes have shown are brought about. So use these words in a description. So you basically have to explain, you know, the changes that you see over here and here. You have to explain it over here using these specific terms: ciliary muscles, lens, and suspensory ligaments, right? So now you can say the ciliary muscles contract, which narrows the diameter of the circular muscles. This loosens the tension on the suspensory ligaments, which allows the lens to revert to its natural rounded shape. The lens now refracts light more strongly, bringing the diverging rays from the nearby object to a focus on the retina. So as you can see, this is actually the normal shape of the lens. So what they, in reality, they just want you to explain what happens to the changes in what happens to changes in the ciliary muscle, the suspensory ligaments, and the lens when the object is at a closer distance. So the ciliary muscles contract and narrows the diameter of the circular muscles, okay, loosen the tension in the suspensory ligaments, which allows the lens to revert back to its rounded shape. The lens now refract light rays more strongly, bringing the diverging rays from the nearby object to a focus. So all around, you could say this question is at least worth 4 marks, because 1 mark is always given to this. When the object is close, ciliary muscles contract, narrow the diameter of the circular muscles, 2 marks. Loosen the tension in the suspensory ligaments, which is 3 marks, which allows the lens to revert back to a rounded shape. The lens now refracts light more strongly, is another 4th mark. So almost, I could say, 3 to 4 marks. So this is actually very important, because they, like 95% of the time that they usually ask questions from the I section, from the I topic of this chapter. They usually ask about what happens when an object is brought closer, what happens to the ciliary muscle, what happens to the circular muscle, what happens to the lens. So it's a very popular question actually. So I'll mention it here. So it's almost worth, you could say, 4 marks. So accommodation in the eye is a reflex action. So what is meant by reflex action? A fast automatic response. Basically, your brain doesn't have to be involved in this reflex action. So in reflex action, there is no involvement of your brain. It's just done. I mean, there is actually involvement of your brain sending the impulse, but it's done so quick that your body knows what has to be done. Okay, and that's always response to a stimulus. That could be a light, a sharp object, anything. So just what could be the stimulus that brings about this reflex action? Now they're asking, what is that specific stimulus that brings this reflex action when the object is brought close to the eye? So remember over here they mentioned about the definition of the stimulus. Well, not stimulus, it was 1 second. Yeah, they mentioned about the types of stimulus. You have light, sound, touch, temperature, and chemicals. So these are actually some types of stimulus that your sense organs respond to. But in this case, it's going to be the blurred image on the retina. Because in the picture over here, if you see, light is focused on the retina. So if light is focused on the retina, it will bring about like a blurred image, because that is what your sense organs, the eye, they're actually responding to. Right, so now in exercise 13.3 you have the Auxin and the tropism. Right, so for the line graphs in this exercise, you're asked to draw best fit lines. Your line should be smooth and should have roughly the same number of points above them as below them. Start both lines exactly at 0, 0, but they do not have to go exactly through the final point. Right, so basically whenever you're drawing line graphs in your biology paper 6, you don't have to always start your graph from 0. It's not like maths. Whatever you plot your points on your graph, the lowest point, you start from there. If your lowest point starts at, like, you could say 2, 5, so you start your line from 2, 5. You don't have to go from 0 to 2, 5. No, that is not math. It's just like this, and it's much more clear in that sense. So a plant growing in a pot was placed on its side in conditions of uniform light. The diagram shows the appearance of the plant after 3 days. This response is known as negative gravitropism. This response is known as negative gravitropism. So explain what is meant by negative gravitropism. So now I recall that early in the video I said that gravitropism is what? Responding to gravity. So if it's responding to gravity, so one kind of definition that you could create on your own about saying negative gravitropism is a response in which part of a plant grows away from the direction in which it is pulled by the gravity. So negative gravitropism is when the plant is growing away from the gravity. Like this whole thing is negative gravitropism, because it's not growing down. What grows down is actually the roots. Wait, give me a second. I'm not able to find that. Okay, but anyways, when you're right. Oh wait. Right. So. Right. So these are the roots. Just imagine these are the roots. So so this one, negative gravitropism, is this. It's growing away from the direction in which the gravity is supposed to pull. It's not responding towards the gravity, but the roots are. But in this case, they're actually asking you to define negative gravitropism for this whole plant that's going away from gravity. So you just say a response in which part of a plant grows away from the direction in which it is pulled by gravity. So just remember, negative meaning it's not growing in the direction in which gravity is pulling it. It's growing away from in the opposite direction. And positive gravitropism meaning it's growing in the direction that the gravity is pulling it. For example, the roots of a plant. Now, part B. A scientist measured the concentration of auxin in the upper and lower surfaces of the plant shoot. She also measured the percentage increase in length of the upper and lower surface of the plant shoot over a period of 1 hour. So now the table shows the results. So now you just need to draw a graph over here, a line graph of best fit lines of each set of results. So on the grid, draw line graphs to show the results in the second table, which is technically this, the one showing percentage increase in the length. Draw both lines on the same. So now you guys might be thinking, how do I draw these two sets of data on the same graph? It's actually quite simple. This is one sample of it I could show you over here. This is one sample of it. I know you won't be seeing this that clearly, it's kind of chopped off actually, but what I want to say, this is one kind of sample. So if you see over here in the question, this thing, time minutes will go on the x-axis because this is independent variable. Independent variable goes always on the x-axis, and the dependent variable, the one that's changed in response to the independent variable, always goes on the y-axis. So this is how it is shown over here. Let me just see if I could bring this. Okay, so anyways, if you see over here down, you have the time, minutes. So don't forget, if the question mentioned minutes, you mention minutes. If it's mentioned in seconds, write seconds. Okay, don't just assume, and don't write short units. If the data or the title mentioned in the graph is like time/s, time/m, only write that, because they're very specific on what you write. They can actually deduct marks by not writing the same thing what they expect you. And then you have on the y-axis percentage increase in length. You just literally only have to copy paste the text from here and write it on the graph on the y-axis. So it's a very simple it's quite a simple one actually, right? So if you see over here, you have 0, 10, 20, 30, 40, 50, 60, 70, and this is time, 10, 20, 30, 40, 50, 60, right? So you just write that exactly over here, over here on the x-axis, and then the percentage increase right down over here. So this is how it looks like. If I zoom it over here, you guys might be able to see this. This is how it is, the whole graph. This is the key and this is the thing. So right, so you just plot the points one by one, okay, and then draw the best fit. So best fit line, in a best fit line, the line does not have to necessarily touch all the points you've plotted. Like you see over here, 1, 2, 3, 4, 5, 6. There are 6 points that are plotted over here, but out of the 6 points, only 5 of them are actually connected, and this one is left out. So it's not a big deal. I mean, it's okay because it's about a best fit line. Until and unless they ask you to draw a line graph, then you have to connect all these lines one by one. But since they asked you a best fit line, you just have to find the best fit. And the same thing goes with the second set of data, which is this one actually. This is the second set of data on the lower surface. So you just do it. So now for the key, normally it's actually much more accurate if you always draw in the X form, like you see it's plotted like this instead of the dots. But if it's like 2 sets of data given to you, then you can also draw it over here. Like if it's X over here, X, and then to differentiate it, you can actually draw X circle around it. So the data is still accurate, the points plotted, the best fit line is also accurate, so it won't affect the accuracy of your graph, right? So let's go on to the final part. Use the results in both tables to explain what made the plant shoot grow upwards after the pot was turned onto its side. So the explanation is there was more auxin on the lower surface than on the upper surface. This made the cells on the lower surface get longer than those on the upper surface, so the shoot curved upwards. What made the plant shoot grow upwards? So you see over here, the plant shoot, it didn't grow down, it didn't go sideways, it grew upwards still as a normal plant. Why is it like that? Is because there was more auxin on the lower surface of the plant, more auxin on the lower surface of the plant than on the upper surface. This made the cells on the lower surface get longer than those on the upper surface, so the shoot curved upwards. So you know, if there is auxin concentration on one end of a plant, then the other end will grow in response to that. So for example, like this is the upper surface. You can't really see the lower surface over here of the plant, but the lower surface of the leaf over here, you see what they're trying to explain is that there were more concentrated auxin on the lower surface of the leaf, or you could say lower surface of the plant, including all the leaves. And so when they actually more auxin on the lower surface, that will actually cause the plant to grow in the opposite direction, meaning it won't grow down. It won't grow. Since it's lower surface, you might be thinking, why is it not going down? But this is the ideology when it's talking about auxin concentration. If auxin is on the lower side, or if it's on the left side, the plant will grow on the right side, or it will grow upwards, right? And if you see the data over here, in the upper surface there was 0.9, 1.1, 1.5, but in the lower surface there was actually more. So what you can conclude is that because there was more concentration of auxin in the lower surface, this is what caused the plant shoot to grow upwards, as there was more concentration of auxin on the lower surface than the upper surface. So this is how you can write in simple terms. So we are actually done all for today. And if you guys have any questions, or if you think I've mentioned something wrong, you can guys let me know in the comment section below, so that I could pin your comment if I deemed it necessary to. And right, stay tuned for the next chapter. Alright, see you.